How does a compound bind to its target protein? My name is Annette Roos. I work with biophysical methods for drug discovery. I'm a crystallographer and structural biologist by training. So how can structure determination help the drug discovery process? I use a method called X-ray crystallography. Here we want the target protein of interest to be crystallized, to be able to visualize the atomic structure. We would like to crystallize the protein target together with the potential binders, the compounds that bind to it. We use 96 well plates with 3 little wells where we put our pure protein solution at pretty high concentrations. We have a crystallization robot to do that. It has tiny little pipettes here that will dispense small volumes. And then we aim for this. The protein should crystallize. We fish out the crystals with a tiny little nylon loop, 100 micrometers. And then we plunge it into liquid nitrogen to preserve it so it doesn't dry out in the air. Then they're sent to the synchrotron. The crystals are put into the X-ray beam. They diffract, and we measure the diffraction pattern. The diffraction pattern is integrated and scaled together. We have to solve something called the phase problem, and then we can get the electron density. And this is the data that we have to be able to determine the protein structure. There's a lot of maths in this step too. And then this is the problem. We have to fit the amino acid sequence of the protein that we have purified and crystallized into the electron density. And that then tells us what the protein looks like on atomic level. And here you can see I have placed phenylalanine into this electron density, and here's a tryptophan. And then this is what you tend to see in pictures of proteins, often ribbon or cartoon representation from blue to red. So blue is the N-terminus, red is the C-terminus, which is there. And then here we have a compound bound in the binding site. And what do we want protein structure information for? Well, we can design better drug molecules, better compounds, if we know how they bind. We can also get information about target engagement. Are the compounds we have that have given a hit in some kind of other assay setting, how do they bind to the target? Where do they bind? If we have antibodies as our drug molecule, then we can also do epitope mapping with X-ray crystallography. So there we get the structure of the antibody together with the target protein. That's visualized here. So here we have a Fab binding to the antigen, and then the epitope is the orange part, the interface between the two molecules. We can also do virtual screening. Then we look at the binding site. We take away anything that is bound there, leave the amino acids, and see what other types of molecules could fit in the same pocket. In structure-based drug design, we zoom in on the binding site and look at the compound that's binding and which amino acids are binding to it. And then from there, we try and change the molecule to get better interactions or more interactions with the amino acids in the binding site. And here, this picture shows the amino acids are in yellow, and then there are 3 compounds that are overlaid here. So it's 3 different structures that have been solved. You superimpose them, and then you can look at, are there any differences in how the amino acids are? In this case, there wasn't. And where do these molecules bind? Where are the differences in the molecules? And here's another way of showing this. We have the protein here, and then we zoom in on the active site here. And that's what the compound looks like when it's bound. This is a kinase, and this is sunitinib, a very well-known kinase binder that's bound here. Then we can see which interactions. Here we have interactions that are called the hinge binding region. Here I would like to first show what the electron density looks like. This is an electron density map of about 2.3 angstrom resolution. And here you can see the blue mesh is the electron density, and the yellow sticks here are the amino acids that have been placed into the electron density. So here you can really appreciate how well we can place the amino acids into the density. Here is an alpha helix, comes along there. This is a so-called graphics program that is visualization of the electron density, and it's three-dimensional, so you get a feeling of the depth. You can see things that are in the front and behind. Here is another graphics program that is more focused on the visualization of the protein. Looking at the electron density and the amino acids in the stick type representation or line representation can be quite, doesn't give you the full overview. So here's the same kinase colored the same way in this rainbow representation, again cartoon, from blue, the N terminus, down to red, which is the C terminus. And here is the binding site for this compound that is called sunitinib. We can zoom in there, and then here you can see more of the depth when I scroll away information. You can see more details. And here now some of the amino acids I have made into the stick representation, so you can see them more clearly. The other amino acids are now hidden. I can switch them on here. Now they're shown as lines instead. And I have highlighted amino acids that are actually in the binding site as this fatter stick representation. Then we can look at the interactions to the compound that we have crystallized this protein with. And we can try and see where can we grow the molecule to achieve maybe more hydrogen bonds. We can also visualize the hydrogen bonds like that. So here you have this hinge binding moiety. So you have hydrogen bonds here to what is called the hinge in a kinase. I can also read in a similar kinase, which I've done here, and overlay it. And then I can compare the binding site. Again, this is sunitinib bound in this binding site. I can compare what amino acids are interacting with sunitinib in this kinase. In general, they will be very conserved. Many residues will be the same, but you can find some differences. And then you can try, if you want to be selective towards a certain kinase, you can try and use the places where the two proteins are different. Where are the amino acids different? Try and grow out your compound so that it can interact with the different amino acids. Then you can achieve selectivity. So at the DDD, we tend to, if possible, crystallize our target together with the compounds that we produce, the small molecules that we develop. But we can also use all the information about structures that is available in the Protein Data Bank, the PDB. So this is a data bank containing thousands of solved structures where the scientists have published a structure and then deposited the coordinates and the data into this database. So I can download a structure from there and the data, check to see that I agree with what has been described in the articles, and then I can use these structures in my drug design as well to get input for how molecules should bind, or just information about the structures that we're working on, the targets that we're working on, rather. So in the PDB, the structures there are based on experimentally recorded data. From the PDB, we've all heard of AlphaFold. So AlphaFold is a prediction program for determining protein structures, predicting protein structures from the amino acid sequence. So we can also use the models that are available by AlphaFold to get an idea of what our targets look like if there is no structure in the PDB database.